Welcome to ModPath Chat, the official podcast of modern pathology, featuring interviews with authors and experts on the latest science, technology, and developments in the field of pathology. Our host, Dr. George Netto, is the editor-in-chief of Modern Pathology and professor and chair of pathology and laboratory medicine at Perelman School of Medicine at the University of Pennsylvania. Here's Dr. Netto.
Welcome to ModPath Chat. My guest today is a dear colleague and friend, Dr. Lisa Rooper, Associate Professor of Pathology and Oncology at Johns Hopkins. Lisa, I can't believe you're already an Associate Professor. I'm so proud of you. Dr. Rooper, not only she's an Associate Professor, is the Acting Director of Surgical Pathology and the Director of Head and Neck Pathology at Hopkins. Clearly, her interest is head and neck and neuroendocrine, and she therefore co-authored many of the chapters in the fifth edition of the WHO Blue Book on both topics. She's also, most importantly, a member of our prestigious editorial board at Modern Path. So she's here today to discuss with us a very rare entity. Looking at the list of co-author, it's almost who's who in head and neck. So we had to put all these co-authors to get to 23 cases. So the entity is sinonasal olfactory carcinoma. So we'll talk about that in a second. But thank you, Dr. Rooper, for accepting my invitation.
Well, thank you so much, Dr. Netto, for inviting me. I'm delighted to be here.
All right. So let's first start with the usual question. Is besides that this is very rare, I realize there's a little bit of a controversy in do we put this in the classification or not? Take us back, and how did the term come about, and what are we talking about here?
Yeah, no. So this is a tumor that people have been aware of for a long time, but historically has just not really ever formally been recognized. So in the sinonasal tract, we sort of think of neuroendocrine tumors similar to how we do in other organs, that there's epithelial neuroendocrine neoplasms such as small cell carcinoma, large cell neuroendocrine carcinoma, generally have keratin positivity as well as expression of neuroendocrine markers. And then there's kind of more neural type neuroendocrine neoplasms. And in the sinonasal tract, this is usually olfactory neuroblastoma, which really are thought to be more of neural lineage. They have S100 positive sustentacular cells. They're negative for keratin. They have a lot of neurofibrillary stroma, rosette formation. So they're sort of recapitulating the developing neural tissues. And these have always been thought to be very separate groups of tumors. But there's always been a group of tumors that sort of meets in the middle. Tumors that sort of have the neural features that look like olfactory neuroblastoma with the neurofibrillary matrix material, lots of rosette formation, but also have really well-developed epithelial elements. So they're diffusely positive for keratin. They have intermixed gland formation. And no one has really known what to do with these tumors in the past. People have put them in a lot of different categories. Some people have said, olfactory neuroblastoma can expand to include these types of tumors. Other people have said no, that they're carcinomas or that they're mixed carcinoma, olfactory neuroblastoma. And one term that has been used is olfactory carcinoma for them. It's been, even though it hasn't really shown up in the literature a lot, Dennis Hefner from the AFIP was one of the first people to advocate for this term. He actually used it in his diagnoses and consultation practice for years. Mm-hmm. And it sort of floated around out there. Although there's never actually been a large series published on these or really had them recognized in tumor classifications until the last couple of years.
Wonderful. So I like that, the mixture that you described, epithelial and neuronal or neuroblastic. So just to make sure, these are true glands, different than true rosettes, and they can have, the lining can have even cilia, as I'm sounding very smart because I read the paper. And actually, I remember at Hopkins, we had few of these when Justin and Dr. Westrow were still there. So I guess I still remember a couple of things and such.
They're they're the kind of things that are problematic that they, come up and are shown at conference and no one knows what to call them. And there's definitely, I always feel like glands versus rosettes seems like it should be really obvious. These tumors definitely have structures where it can be challenging to tell which is which, but you definitely have things on both sides. There's glands, they do sometimes have cilia, they have mucin formation, mucin production, and then on the other side, you really do have rosettes that have some, neurofibrillary stroma involved with it, so that, that are truly neural structure. So they really are, both elements can be there.
Wonderful. So I guess the main purpose of the study is that you collected 23 cases. I mentioned that before, and for molecular studies. So tell me just briefly what kind of studies you did and what are the salient findings.
Yeah, no, absolutely. And this is actually our second paper on this entity. Our original paper, which was published a couple of years ago, had been basically a histologic descriptor, since this had never been well characterized in the literature. And we had, despite how rare they were, we'd had more than 50 cases for that study, because it's the kind of thing where you ask anyone in the head and neck pathology community if you've ever seen this, and they're like, yes, I have a couple. I never knew what to do with those. So it's really kind of a point of tension in diagnosis that people have not known what to do with them. So, the previous study, we basically characterized the histologic features that I just described. So now we wanted to take it a little bit further and see if there were molecular commonalities among these tumors too. So, in this study, we were restricted to just over 20 tumors that we had recent tissue blocks available that we were able to do next generation sequencing on. So we did perform next generation sequencing using a large custom panel on all cases, and we also were able to gather immunohistochemical results, filled in a few stains, but largely resulting from the diagnostic workup on all of these cases, since these are usually cases that are stained widely as part of diagnosis.
Excellent. And what were the recurrent alterations?
Yeah, so we actually found that there were two main groups of recurrent molecular alterations in olfactory carcinoma. One is Wnt pathway alterations, most commonly CTNNB1 or beta-catenin gene mutations. So that was the most common alteration. And then another subgroup of tumors had ARID1A alterations, which is, of course, a member of the SWI/SNF protein chromatin remodeling and complex pathway. So those were kind of the two largest groups. And then there were a couple smaller groups. There were a couple cases that had IDH2 mutations that morphologically fit in the olfactory carcinoma spectrum. And then another group that had RUNX1 alterations, which is not common in solid tumors, and we're not entirely certain what to do with.
Wonderful. So we'll come back to the significance of the IDH2. But on the immunohistochemistry side, beside, you already mentioned the centicular cells, the cytokeratins, something about beta-catenin?
Yeah, so they are, so they're definitely keratin positive, and diagnostically speaking, this is one of the most helpful features for recognizing them, because even though they look a lot like olfactory neuroblastoma, they will have keratin positivity that's not focal, often pretty diffuse, especially using a low molecular weight keratin like CAM 5.2, generally some expression of pan-keratins like AA1, AA3 as well. Most cases have S100 positive sustentacular cells. It's usually not around every single nest of the tumor, but if you stain a section from the tumor, you will see areas. And about half the cases, it's interesting, sort of have P63 or P40 positivity in a basal distribution, which is an unusual pattern that, it's not clear what it means. It's definitely in kind of the neuroblastic cells, not in the epithelial cells, but it's there. And of course, they are positive for neuroendocrine markers at variable intensity, but you should see some degree of synaptophysin, chromogranin, INSM1, good specific neuroendocrine markers. The beta-catenin expression is variable. We weren't able to perform beta-catenin on all the cases that had the CTNNB1 mutations, but a subset of them did have nuclear beta-catenin positivity. It was not all of the cases. It seems to be a little bit variable on the protein expression side, but all the molecular alterations were, in loci that have been characterized as oncogenic in the past. So, we feel like these are oncogenic drivers regardless of what's happening with the protein expression.
Mm-hmm, mm-hmm. And I refer the audience, the listener, to Figure 4, where you're showing a lot of these immunohistochemicals, but it's striking this rimming of the P63. So you're saying it's not myoepithelial, it's just truly in neuronal or neuroepithelial cells.
Right. No, it's not. It doesn't stand for any other myoepithelial marker. It's different cells than the ones that are standing for S100. It's something in those, cells that have that primitive neuroectodermal differentiation that are expressing the P63 and P40. So we don't know why it's doing that yet.
So I guess clearly the problem with these, if you do truly have the two component, very obvious, but if you end up with predominantly the neuronal, you can see few cytokeratin positive cells, you question yourself. So the differential does still include OMB, olfactory neuroblastoma and other things. Put it into context. So how does the molecular help in your daily practice? Do you do that, or you anticipate that now that you know what are the recurrent alterations you're going to help, or actually...
Yeah, no, I think it probably is not something we need to do in daily practice. I think, at least right now, it was very interesting academically. I think, the big takeaway in my mind from this paper is, these have a characteristic histologic and immunohistochemical profile, and they have recurrent molecular findings too. So I think we've, kind of helped solidify that there's recurrent features of this group of tumors that was previously pretty poorly characterized. But the molecular findings are not entirely specific. There's other tumors in the sinonasal tract that have CTNNB1 mutations, other tumors that have ARID1A alterations. So they're not entirely specific. It doesn't entirely help differentiate them from olfactory neuroblastoma. Olfactory neuroblastoma actually is still a really weird group of tumors in that there's not a clear recurrent molecular alteration. There's not anything diagnostic that we can do for that, which is so rare in the sinonasal tract as everything's sort of becoming molecularly defined. So, finding these recurrent alterations, they're not really there recurrently in olfactory neuroblastoma, but, it's kind of an ill-defined area, so it's hard to lean on that entirely. So really this is a histologic and immunohistochemical diagnosis that we all can make without the help of molecular at this point. I think we've just learned some more about the underpinnings and possibly the relationships.
And anything about the IDH2, because I see in your conclusion you talk about a spectrum now. Maybe look at that. Can you explain to me a little?
So IDH2 mutations were originally discovered in sinonasal undifferentiated carcinoma, kind of the high-grade sinonasal carcinomas that aren't staining for anything else, kind of after all these new molecular categories have been excluded. They were then subsequently reported in large cell neuroendocrine carcinoma, so tumors that have neuroendocrine differentiation in the sinonasal tract. So it's sort of understood that this IDH2 mutations can be polyphenotypic. There's actually been some controversy about IDH2 and olfactory neuroblastoma. There have been a few papers that have said IDH2 mutations can be seen in olfactory neuroblastoma, and then other papers have said no, these tumor types are better classified in other categories. And really probably what a subset of them are the ones that meet this criteria for olfactory carcinoma, the neuroectodermal type of tumors that have epithelial differentiation. And there's been a push over the last few years, led by Snehana Dogan and colleagues at MSK, to start calling IDH2 mutant sinonasal carcinomas as a separate category. So regardless of whether they're undifferentiated, neuroendocrine, neuroblastic, like whichever category you want to put them in, the thought is now that molecular alteration should supersede anything else. And I think that's going to be the simplest thing in terms of classification, that we can just, defer to IDH2 mutation defines this entity, and that can supersede anything else. And after that, we can look at the histologic and immunohistochemical characteristics of what is left, and anything, that meets the criteria after excluding IDH2. Those are what I would call olfactory carcinoma down the road.
Fascinating. It's, I guess it's close enough to the brain that you can rely just on the molecular and then figure out the morphology.
Yeah, no, that's what people want to do, right?
But unfortunately, it didn't work here. But nevertheless, it's still a fascinating study. And I know I learned a lot from it. So thank you for taking the time from your busy schedule to do this podcast. And I look forward to more of your work and your esteemed colleagues' work to come to the journal. And thank you for everything you do for the editorial board as a member.
Of course, thank you again for the invitation.
Any opinions expressed in this podcast are the speaker's own and do not represent the views of Modern Pathology, Elsevier, the University of Pennsylvania, or USCAP. Your ModPath Chat host and scientific director is Dr. George Netto. The producers are Dr. Catherine Ketchum and Carly Hess. Technical direction is provided by Kaminsky Productions. Music by Mitch Neubauer. Thanks to the authors, reviewers, and editors of Modern Pathology for making this podcast possible.